Today you're going to be taking Cornell notes about cells. There are two main types of cells. The first ones are eukaryotic cells. Eukaryotic cells are cells that contain a true nucleus. So they have organelles that are surrounded by membranes that have specific functions. So they have a true nucleus. So the genetic material is enclosed within a membrane. All complex cells, such as plants, animals, protists, and fungi, including us, are made up of eukaryotic cells. The other type of cell is a prokaryotic cell. Pro, the root means kind of primitive or before. So these are cells that do not have a true nucleus. So their genetic material is free floating within the cell. They don't have any membrane-bound organelles as well. And examples of prokaryotic cells are bacteria. They're very simple, and they're also very primitive types of cells. We're going to be focusing now on eukaryotic cells. And the two that we're going to be looking at the closest are plant cells and animal cells. Now, plant cells and animal cells share a lot in common. However, there are some unique things that plant cells have. One is that they have a large central vacuole. You can see it in this diagram. You can also see it in this picture here, this big kind of clear spot. Those are vacuoles. And each cell has one large central vacuole. Now, all cells have a cell membrane which surround the cell. It's this thin line right here. However, plant cells also have a cell wall, which is outside of the cell membrane. And the cell wall helps with giving structure and support to the plants. Plant cells also contain chloroplast. It's an organelle that contains a pigment, chlorophyll, and it is where photosynthesis takes place. Now, many people think that plants go through photosynthesis, animal cells go through cellular respiration. Plants do both. They do photosynthesis using chloroplast. They also go through cellular respiration. So plants do have mitochondria. That's not a difference between plant and animal cells. They both have mitochondria. For this drawing, you should have a handout, and I want you to label all of the different parts of this cell on your handout. That handout will then be glued into your notes. Next are animal cells. Animal cells do not have a cell wall. They just have a membrane surrounding them. Animal cells also contain mitochondria, but they do not have chloroplasts. Now you're going to fill out the different organelles of this animal cell diagram and glue this into your notes. Okay, so now we're going to look at the structures and functions of cells. Every cell has structures that help it carry out the functions of life, and they are called organelles. So organelles are tiny specialized structures that have a particular role inside of each eukaryotic cell. Now, organelles are in eukaryotic cells, not in prokaryotes. All cells, prokaryote and eukaryote, have cell membranes. The cell membrane is the boundary of the cell, and it is semi-permeable. Permeable means something being able to go through it. So semi-permeable means that some things can go through a cell membrane and some things can't. So the cell membrane regulates what goes in and out of your cell. It also holds everything together, and it helps maintain homeostasis. Because if it only allows certain things in or out, it's helping keeping an internal balance within the cell. And sometimes you might see a cell membrane being referred to as a plasma membrane. A plasma membrane is just another way to say cell membrane. And one of the other cell structures is a cell wall. And cell walls are found just in plant cells, not animal cells. They provide structure and support for the cells, and it lies on the outside part of the cell membrane. Now we're going to look at some eukaryotic organelles. Here is the nucleus, and the nucleus is where genetic material is stored. And it also has another little part inside of it called the nucleolus. And the nucleolus is where ribosomes are produced in a cell. Now, ribosomes are these small circular-like dots that you kind of see floating around. When they're floating around, they are kind of floating freely in a cell, they'll just look like these little dots. And these are molecules that are responsible for making proteins. Now, there's something called the endoplasmic reticulum. Endoplasmic reticulum is an organelle, and there's two different types, rough ER and smooth ER. Here you can see that along the endoplasmic reticulum, there are little dots. Those are the ribosomes. Here, they're not free-floating, they're actually attached to the endoplasmic reticulum. So this is an example of rough ER. The ribosomes attached to it make the surface of the ER rough. And the rough ER is where proteins are produced, in particular for export out of the cell. Next, we have smooth ER, and you can see there's no dots along the outside of it, so it has a smooth appearance. And the smooth ER is where lipids are produced, and it also helps with detoxification within your cells. The next organelle is the Golgi apparatus, and the Golgi apparatus takes the proteins that were produced in the rough endoplasmic reticulum, and it packs them up into a membrane-bound vesicle, like a little container, that it sends to the cell membrane to potentially go out of the cell to other parts of your body. Next, we have the mitochondria, and the mitochondria is the site where cellular respiration takes place. This is where glucose is converted into ATP. ATP is the energy that your cells need to do all their different functions. Next, we have peroxisomes. They destroy lipids. They break them apart into their smaller pieces, and they have enzymes there that help them do that. Next is lysosomes. Lysosomes help break down or destroy proteins. And then we use the parts of the proteins, the amino acids, to build other things, and we can reuse them. Next are three different types of tubing within your cells. The cilia and flagella are examples of microtubules, and they're the biggest types of tubing or structure support in your cells. They're involved in cell movement, and they help provide pathways for secretory vesicles to move through the cell. And they're even involved in cell division as they're a part of the mitotic spindle, so during mitosis when homologous chromosomes get pulled apart. We'll learn more about this later this school year. Next are the medium-sized or intermediate-sized tubes or filaments. And they're very stable, and they help provide structure to the nuclear envelope and anchor organelles also through the process of mitosis as well as meiosis. And then lastly are the smallest filaments or strings, and these are microfilaments. The specific example are actin filaments, and they help with cell movement such as actin and myosin, which help in our muscles moving. Next we have chloroplast, chromoplast, and amyloplast. These are basically organelles that store different things. So chloroplasts store green pigment, chlorophyll, and it's only in plant cells. It's a site of photosynthesis, and it's where light energy is converted into glucose. Chromoplasts look basically identical to chloroplasts, except chloroplasts are green, chromoplasts are other colors. And so they are plastoids which synthesize and store pigments. If they're orange, it probably is a carotenoid. So carrots, that would be the pigment in a carrot. The next one are amyloplasts, and these are non-pigmented organelles found in some plant cells, and it's responsible for the synthesis and the storage of starch granules. So this one here is what an amyloplast looks like. Then the last thing that we have in plant cells are the central vacuole, and it's often the largest organelle within the cell, and it holds materials and wastes, and it helps maintain the proper pressure so that the plant cell can stand upright and have the structure and support for growing. Now, to review, what I'd like for you to do is draw this Venn diagram in your notes. And then you're going to use all these words or phrases down at the bottom, and you're going to place them in the correct spot. So if it's prokaryotic only, you put it here. If it's eukaryotic only, you put it on the right side. And if it's something that both prokaryotes and eukaryotes share, you'd put it right in the center. So go ahead and press pause and work on creating your Venn diagram. You're going to create a second Venn diagram now, and you're going to place animals, everything that animal cells have on the left, What plant cells have unique to them on the right, and what both animal and plant cells share will be in the center. So go ahead and press pause and work on your animal and plant cell Venn diagram. Here are your answers for prokaryotic cells and eukaryotic cells. So if you missed anything, go ahead and fix it and make changes to your Venn diagram. Here's the plant and animal cell Venn diagram answers. So go ahead and review your work and make any changes that you need to. Now you are going to need to write your questions and your summary for your Cornell notes. You should have a minimum of 5 questions and a minimum of 5 sentences for your summary. Now in your summary, you should have facts and details. Don't just say that you learned the difference between animal and plant cells and prokaryotes and eukaryotes. I want to know what specifically you learned. So you should have details and facts from your notes within your summary.